Welcome to Science Matters. In this episode, we will be exploring the eye beyond sight, and we will be finding out how we see images like this on a regular basis. Here we can see a normal person undergoing many stages of automatic reflexes as a result of electrical message signals relaying from the brain. However, we want to look more closely at the finer details of the eye. The eye. It is made up of many complex structures, ranging from muscles to ligaments and nerves. Looking more closely and magnifying the area around the fovea, we can see what is actually going on. This image is a zoomed-in version of the structure of the retina. Upon closer observation, we can see that there is a layer of light-sensitive cells, or photoreceptors. One type of photoreceptor which we know about, and need to learn about in this episode, is the rod. We have around 120 million evenly spread rods in each eye across the retina. Rods are a very basic photoreceptor and only provide a black and white picture, which is very useful in low light intensities or at night. Rods are made up of four regions. Firstly is the outer segment. This is the light sensitive region where light energy acts as a stimulus to the production of generated potential. It contains flattened membranous vesicles filled with rhodopsin, which we will look at in detail later on. The second region of rods is the constriction zone. This is a narrow region connecting the outer and inner segments. It is lined with two cilia, which have a rather atypical structure. Next we have the inner segment. This is packed full of mitochondria, which produce energy for the various processes. The cell nucleus is also located here. And finally, the synaptic region, which forms the synapse between its own cells and bipolar cells, allowing electrical messages to be sent to and from the brain from the rods. The sensitivity of the rods is based on the reaction of the visual pigment rhodopsin with light. Rhodopsin is made up of opsin and retinal, and is key to events leading to sight. When light hits rhodopsin, it changes the retinal shape, breaking up into its core components. This stage is known as bleaching. Usually, the cell's membrane's permeability to sodium ions is rather in favor, compared to most neurons anyway, which allows sodium ions ease of access into the rods. When bleached, however, this permeability is lost, triggering a cascade of reactions. Sodium is continually pumped out as normal, but no sodium is entering the cell. This leads to hyperpolarization, causing a generator potential. A message is then sent to the brain via the optic nerve, which actually generates sight. Once bleached, rods are unable to be re-stimulated. ATP energy is required for rhodopsin to be reshapen so that the visual sight process can be repeated. ATP energy is produced by the many mitochondria found in the inner segment of the rod.